All right. Hello, everybody. It's Chris Frew with BioBuzz here for another edition of Ask Me Anything. This time, I'm really excited to have Charlie with me, who is an expert in flow cytometry. Charlie's going to talk about careers in flow, but also careers in science in general, and really share his expertise and his observations with us about of his career that has evolved in the science field. So Charlie, welcome today.
Thank you so much for having me. I'm very excited to be able to come here and talk about this with you guys.
Absolutely. Well, we're doing these every week, Charlie, and I'm excited to have you on. So our goal with BioBuzz is really to create more buzz around science careers. And
Yeah,
as we know, there's a lot of different career paths people can take, right? I mean, they can go a lot of different directions. You started off and on the bench, and then more recently at Slingshot, you were one of the founding members of the commercial team. So now moved into a sales role, and so that's one path that I'm sure you're going to share some observations. But at BioBuzz, we want to start talking about all the different careers and be a
yep,
place and a resource people can come to and say, hey, what do I want to do next, or what do I want to get into?
absolutely. And I can definitely say now in my role, especially, there are a lot of different things you can do out there that may not necessarily be at the bench, where you are still very much intellectually engaged in the scientific process. Whether it is sales roles, writing, strategy. I've talked to so many people with so much knowledge that are doing what, when I was going through grad school, I never even realized existed as far as, influential work.
That's great. And I think a lot of people still don't. So hopefully today's conversation will be a little bit of a taste of that, and
Yeah.
open some other people's eyes, and maybe the next person we have on a year from now will talk about their career that started today when you inspired them to take the leap off the bench.
Oh, I sure hope so. That would be
So
very
before
cool.
we, before we jump into your presentation, Charlie, I just want to take a second to thank BioTrack, one of our sponsors for today. BioTrack provides hands-on workshops on some of the most advanced research techniques. They're located as part of Montgomery College in Maryland. So if you're looking to advance your research skills and everything from flow cytometry to R programming or CRISPR or single cell RNA-seq, they've got the workshops and the resources for you to really increase your scientific prowess, if you will, and make sure you're advancing your career. So thank you again to BioTrack. They've been around for many decades, have trained over 18,000 scientists, so you're in good hands. So with that, Charlie, I'm going to give it to you to take it over. I'm going to turn your presentation on, if that's okay.
Yeah, please do. All right, look at that. That's slick. So again, I'm very grateful to be here today to kind of give this little 10-minute spiel, essentially. And the ask was to talk about a little bit of my career, a little bit of what it looks like to be in a sales role, and then also something that would be kind of from the technical side useful to people. So that is in 10 minutes, a pretty tall ask. And what I wanted to do is kind of focus it around this idea. There's this buzzword in cytometry, especially called fit for purpose. And so, what is it? And here are my reflections on it. So, the first most important thing I got to say, all of this is my opinion, everything, the ideas, the content, etc. And no one is paying me to say any of this. This is all me. I am not representing Slingshot. It's not an endorsement. It's not an official position. So, what I wanted to kind of cover is, what is scientific sales? How did I get here? What is fitness for purpose? Context and the spectrum of cytometry that I've now observed, and then kind of some key takeaways and resources for folks who are interested in kind of upgrading their flow cytometry. And really, it's probably applicable to more than just flow. So, scientific sales. What is this? I never personally expected to be in a sales role. I was going to go to grad school, get a PhD, become a postdoc, and then start my own lab, and then eventually get into startups. Suffice to say, that didn't happen. But what I realized and what I really connected with are some of the aims that a scientific salesperson or a salesperson scientist has, right? The goals here are identify the need of your customer and then connect them with a solution, whether that is something that my company sells, something that another company sells, or being really creative and working orthogonally. Sometimes it's actually connections to talent in the pool. And I've had whole relationships that progressed because of the people I was able to connect them with and move forward. And so on the sales side, what is fitness for purpose in sales versus marketing or bench science, strategy, regulatory, et cetera? The big things you're looking at here are either a technical background or an ability and willingness to learn, right? It's lack of knowledge in a particular subfield is not prohibitive. You just have to be a self starter and to pick it up and pursue it. You have to be able to very quickly essentialize key concepts and get to the why. Cutting through the detail to understand the core idea that is in front of you and the core need that the scientist you're working with has, and then to potentially zoom it out and look at a whole different array of solutions to meet some of those needs. So this requires a little bit of big picture or strategic thinking where, maybe you get stuck on one track, you got to be able to step back and figure out how to come at your question from another angle on occasion. This is also a hardworking position. If you've made it through academia, if you are in industry, you are probably no stranger to potentially long hours. There is a high failure rate, right? Every relationship, you're going to have successes and you're going to have failures, and you just keep going, is what it is. It's that persistence, just like it is to get to the data. And then finally, you do need to have a level of emotional intelligence. And one of the big things is you have to love people. At the end of the day, you are going to be people facing, and you could spend anywhere from a few minutes to 18 hours a day talking to people, depending on your role and the conditions. So I'm going to move us over now to fitness for purpose in a more scientific context, which is directly applicable to cytometry. And I'm pulling this actually from a document called H62, published by CLSI. I really enjoy it. It's a technical manual, and I think it has a lot of really good details and definitions. And they say, what is fit for purpose? It's a validation that is appropriate for the context of use and the associated regulatory requirements. And that, in our time together, is where I want to kind of focus. So questioning your purpose, right? What does my assay need to do scientifically? What are the limits? How low can I go and feel confident about my measurement? How reproducible is it, et cetera? What are the regulatory requirements? And I'll get into this too a little bit more. Is this something I'm doing under CAP or CLIA? Is it IVD? Am I purely academic research? Is the FDA involved? These are all critical questions when designing an assay. And finally, and I think this one can be one of the scariest, is what are the consequences of poor experimental design, right? And that varies quite a bit, and over here, I can talk about that a little bit more, where you have the cytometry spectrum. And I've played around with a bunch of different ways of understanding the different kinds of cytometrists I've had the pleasure of interacting with over the past couple of years. And I think one of the best ways to kind of think about mindsets is on this spectrum of regulated versus unregulated thinking. On the unregulated side, where you've got academia, industrial research, and things like that, the consequence of a poor experimental design, it might be delays in your project, it might be rework, you may accidentally or unfortunately sacrifice some animals that didn't need to be. And because of that, there is a lot of flexibility. You can grab a new antibody out of the latest and greatest paper, throw it in your panel and see what happens. That is a really, really powerful thing to be able to do. And on the other side, as you move closer to more regulated environments, where we're talking about things like clinical diagnostics, manufacturing, release testing for cell and gene therapies become really big using flow now. The emphasis there is on robustness. You want to know that if you accidentally swap out your PBS for coffee, how that assay is going to perform. And this is kind of a narrow workflow, just to be a little more characteristic about it. You start with research, right? The big ideas, the screening, the concepts. It's very broad. There's a lot of flexibility to dig in. And as you move towards a translational state, maybe a finding is starting to mature into, a nascent startup, or it's an asset that a pharma company is interested in converting into potentially a drug. You're refining that down. And in that process, things do become a little bit more inflexible. They can still change, but they aren't locked in necessarily the same way. And then finally, as you kind of hit manufacture, which could be, I'm defining that as like an end state, right? It could be data from a clinical trial. It could be a cell therapy drug product, right? This is whatever you are producing, you are at the end of the line. This is very narrow. It's specific, it is defined, and very often this information is being shared with or tested by regulatory agencies. So how do we think about each other, right? I came from research. I went into clinical diagnostic assay development at the Children's Hospital in Philadelphia. That was a big shift in my thinking, going from kind of the very flexible thinking of a research environment to a more translational and/or end result oriented, regulated output, where realistically, if you screw up that assay, you do a bad job on it, you could put somebody's life at risk. Those are very different contexts. And kind of where I'd like to encourage everybody to go in the course of this, and what I'm hopefully bringing some awareness to, is that there is a lot of good literature out there for how to design quality assays and to validate quality assays. And this is one. CLSI's H62. It's a tome. It's something like 250 pages, but it goes through all of the details, all of the definitions. And I found it really valuable, and I think it would be valuable regardless of your situation in the cytometry spectrum, because it does rigorously show you what a high quality assay looks like. And so, if you're in manufacturing, this can give you insights into what had to happen from research to get it to your hands. And conversely, if you're in more of a research environment, it can give you some insights into what scientists down the line on your project are going to have to do to actualize those findings. Another really nice one that is under 50 pages is this protocol from Current Protocols. It covers a lot of the same material. The language on it is a little less formal, a little less dry, I would say. But it does also do a really nice job getting into some examples, riffing off that fit for purpose idea. They talk about, what does it look like to validate a research assay? What does it look like to validate a clinical assay, et cetera? And they do it in a really nice, concrete way that I enjoyed. The other two things to kind of make everyone aware of, if you are not already, two wonderful organizations. There's ISAC, which publishes Cytometry Part A, and ICCS, which publishes Cytometry Part B. Both of these are great for kind of keeping an eye on the pulse of cytometry in multiple different domains. And the cool thing about them is they often publish process or method papers that are designed specifically to be picked up and applied to other things. So rather than getting to a new cell type and a specific finding, it might be a methodology for how to transfer assays to your friend in another country. And so kind of the big takeaways I'd like to leave everyone with here: know your context, understand yourself, understand your goal, understand the assay you're being asked to perform, check the literature, because there are people out there who have probably done something similar, if not exactly the same thing. And then design your assay to fit that purpose. And finally, zoom out, remember your big picture. It's all of science is not, you're not on your own island. You are collaborating with other people, whether you kind of are able to realize that or not. Other people will be interacting with your work in some way, shape, or form. And just keep in mind the perspective that they may be bringing to it and the questions that they have. And it can really help speed up transfer processes and actualize discovery into therapeutics. And ultimately, that is one of my favorite things to be able to do in this role. Help people move along, design what they need, and make the process faster so that health generally can improve. And so I have a couple of other papers in here that I found to be very informative. I'm including them not for you all to very quickly try and write them down or screenshot them as fast as you can, but so that these can be referenced in the future as this goes to some kind of blog post. I know, Chris, you're talking about putting that together. So that's my whole spiel. I love talking about this stuff. I'm very into the technical. It's a lot of fun for me, as odd as I sometimes feel saying that. Please feel free to reach out. That's my email. I can also be connected with on LinkedIn. Always happy to talk about it. And I really appreciate the time. That's all I got for you.
Well, that's great. Well, not all of what you got for it, because I got some things for you as well. So,
oh boy. Okay.
So we got some questions. This is an Ask Me Anything, so you never know what's going to come at you. But that was great, and I love the fit for purpose. And I think that at BioBuzz, and hopefully what we're working towards is a career fit for purpose.
Exactly.
Helping people with a career fit for purpose. So I love that element that you're bringing to the conversation here today, Charlie.
Yeah.
So a couple questions for you.
Please.
If I'm at the bench right now and I'm Charlie 5 years ago, what do I have to start thinking about if I want to transition into like a commercial role or off the bench into sales? Like, what are some tangible things that I need to start thinking about expanding my awareness of or preparing for?
So I would say kind of the first and most important thing is to actually understand yourself, right? Figure out that fitness for purpose. For me, one of the things I kept running into in my bench career is that I would get kind of wanderlust intellectually after about a year in any particular role. So it was very hard for me to kind of sit still and focus on one thing for years and years at a time. And I also found myself kind of routinely craving faster paced and higher scale interpersonal interactions. I do enjoy people. I like talking. I like exploring lots of ideas quickly. Um, for me, it was that need for more personal interaction and to be able to talk more broadly about science, to be able to talk with other experts and things like that was really, really exciting and attractive. And then I kind of took stock of like my own abilities, right? I have a theater background, oddly enough, and improv comedy was probably one of the most helpful experiences that I've had for a sales role. Like the sheer power of yes and is it can't be understated. The other things that I was looking at about myself are like I like novelty. I like moving around and doing different stuff every day, and I like having a fast paced environment. And so for me, sales was a really good position for me to get into. I have had the pleasure of working with a bunch of really talented marketing people who have similar technical backgrounds. And what I've observed is that for them, often they're not as much people oriented. They're not looking forward to having 18 straight hours of conversation in a day like I might. They are very much more like kind of strategic and like big picture. And there's a whole lot of creativity that goes into that in a totally different way, right? And those two types of careers are always, I think, in demand. I'm constantly getting bombarded in LinkedIn for technical sales roles, and I see there's always marketing roles available, right? You got to be able to speak the language to really reach your customer base. But there's a lot of stuff out there too that I haven't even interfaced with that heavily, but have had the chance to talk to folks who are into regulatory strategy
Mhm.
is one great example. You have to understand, on the one hand, the character and the science of a drug that you might be putting forth to the FDA. And you also have to be able to speak enough of the regulatory language to craft a strategy, let's say, to get it through FDA approval. So that's another thing. Medical writing, if you don't want to be slinging pipettes anymore, or technical writing, I could say you don't want to sling pipettes. You want to sit quietly at home and click clack away with your cup of coffee and still do science and think. That's another way you can go too. It really comes down to taking stock of who you are and kind of where you want to be. And for me, it took almost 10 years of bench work to really understand what I wanted to do and what was going to make me happy in my career.
Always comes back down to knowing thyself, right?
Exactly.
Which is harder to do than it sounds.
Well, that's why you got to try stuff, right? You read. I was reading tons of different journals, talking to everybody I could. Befriend your sales reps because we're kind of like I probably got more than a few fantasy readers on here, but like there's always the trope of the tinker who goes
Yeah.
from town to town and he's got like all the pots and pans and he'll sharpen your knives and like give you news from two towns over.
Yeah.
Salespeople are like that. We talk to a ton of scientists all the time. And especially in my role, I'm really getting into the details of what they are doing.
Yeah.
And so for kind of more junior folks, I often get questions about like, what can I do in my career? What have you seen people doing? Just like you're saying. And then there's also the technical side of like, I have seen how other people have accomplished what you're trying to do. We can have conversations about that. There's a lot I can't say, but what I do enjoy doing is asking very pointed and specific questions apropos of nothing that eventually induce some kind of thought process. And when I feel like it, I can go out and make life a lot easier for customers.
Yeah, that's great. So a couple takeaways I heard was know thyself, befriend your sales reps if you want to. And I would just say maybe the message there is befriend the people in the role that you want to get into, right? Maybe that's
Absolutely.
the approach. But again, if it's sales, that I'm sure there's salespeople coming by your lab on a regular basis.
All the time.
Ask them to take you to lunch and tell them what you do.
Yeah.
So and then read and be curious. I mean, those are some really good takeaways for it that I'm kind of pulling out of that.
And yeah, absolutely. And also, I'd say looking for professional societies if you have an interest in a particular type of scientific career. There may be a professional society for that. And also just finding people on LinkedIn. If you don't have a LinkedIn, go make one. Start connecting with people. Go to workshops. Connect with BioBuzz. All those kind of things.
Great advice, especially that last one.
Absolutely.
Great. Well, so let's take a step forward also, or a step in, more granular, I guess. Flow cytometry, right? So as a person who places people in jobs, as a recruiter, like I see the flow cytometry a lot as a requirement that companies are looking for.
Yes.
I'd love to hear, why is that growing, or why does it seem to be growing as a tool, or as a skill set that's needed, and where do you see the growth in the area?
Absolutely. So that is really kind of the genesis of where Slingshot came from, just because there is so much growth in flow. And as we've learned over the past couple of years, it's in a lot of different ways. Flow cytometry itself, fundamentally, it allows you to query many different things about many cells very quickly. And that is very, very powerful depending on how you want to apply it. So in the clinical world, for example, these are folks who may have either an MLS or an MLT degree. There is a massive demand for people who understand cytometry to do clinical diagnostic testing, right? Can you read the plots? Can you work the cytometer? Do you understand the protocols and what you're doing? When I left CHOP in 2022, I think it was something like 6,000 MLS grads every year for 24,000 open roles. So demand is extremely high in those worlds. When it comes to kind of industrial cytometry, it really depends on the goal and the asset that you're working on. So really upstream research and development, if you have anything going on related to immunology, oncology, actually a whole bunch of other disciplines now too, it's that ability to design an assay that can really, really quickly get a lot of information out of a lot of cells and then do it reproducibly over time. That is something of an art. And that's much more similar to kind of like the academic research side of things. As you move into translation, the roles there are very heavily focused on using cytometers to kind of develop the robustness of that assay, right? The goal is to take an asset from an R&D environment and to pare it down to the core data that are needed to eventually pass to a clinical trialist or a manufacturing organization. The emphases there are going to be very much on process, right? The things that you'll see, especially if you go to something like H62, it's, we took this assay, we ran it in triplicates with 2 people on 3 machines over 3 days, and then we did it on, 50 different permutations of the following sets of conditions, understanding very, very deeply the limits. And as a part of that, you have to understand the limit of your cytometer and the limit of that assay. Because how could you take something from research if you don't know kind of soup to nuts what's going on? You have to understand the fundamentals to break it down in that way. And then finally, when you get to more like a manufacturer or let's say clinical trial, like if data is your output, cytometers there are being used in a couple different ways. Clinical trials, you can have the big expansive panels that R&D might often see. They've just been qualified to a higher level. And now you have questions of scale, right? Can I take this assay? What does my protocol look like? And can I do it on 1,000 people over the next 2 years in such a way that all of that data can be interrelated across those 2 years? When it comes to things like manufacturing, let's say, or quality control release, if you're working with any kind of cellular therapy, gene therapy, cell therapy, they're kind of, in my mind, similar. Can you express the protein or the construct that you're looking for with the markers that indicate to you and your team that drug product is going to be successful? And so, that picture I put up of like research to translation to kind of manufacturing, to put it in flow terms, you might be looking high parameter in research. You're looking 20 to 50 colors, a lot of different data coming out. Translation, you're going to bring it down to 10. And then finally, when it hits like a manufacturing space, you need the cytometry to be really 4 or 5 colors tops.
Yeah.
And understanding why that is also critical.
That's really good. That's a very nice kind of flow of how you use flow.
Yeah, exactly. And then I should say there's also the folks who are doing flow for the sake of flow, and those are some of the most fun people to talk to. Those are the people who are involved in actually developing new cytometers and new instrumentation And that really, really starts moving quickly out of my own area of expertise and into things like microfluidics, physics, in a whole bunch of different ways.
Yeah.
It gets really cool out there at the edges as well.
Yeah. Yeah. You can tell you're not a flow geek, are you? You're just like,
Nope.
love it.
Zero.
Man. I love it.
Yeah.
Well, that's really helpful. Yeah, to see that. And again, we see that all the time, and we'll see, from people like, I need someone who has, 24 color flow
Yep.
experience. And then sometimes you'll see people like, hey, look, I need someone who can optimize. And like, so again, it's like, we see it across the spectrum.
Yeah, which is another semi-unintended pun. It was intended in my talk. One of the things now is the shift from conventional to spectral cytometry, and the additional details that can come into play as you make that shift, if you should choose to do it.
Yeah.
So an understanding of spectral is another layer on top of kind of conventional high parameter.
Yeah. So tell us a little bit about, if you don't mind, tell us a little bit about
Mhm.
Slingshot. Like, what is, what is Slingshot doing for your customers? First of all, like, what, love to hear that story.
Mhm.
And maybe also you could take that into, just walk through, like, what's a day in the life of Charlie.
Oh boy. Um, I'm sorry. Could you repeat the
The
first part
first
of the
one is, just
question?
tell us about
Yeah.
Slingshot. So,
Okay.
you told us about kind of high level that you took your career there, and high level what you do. But I
You
love
uh,
to hear
you
what it.
scared me with the idea of talking about myself more. Um, so Slingshot, I came to Slingshot after working in clinical assay development, and I was developing assays and validating assays for primary immune deficiencies, where the incidence rate, the most common deficiency I would see was like 1 in 50,000 children, which meant that in almost 2 years in that role, I saw 2 patients who actually had it.
Mhm.
So if you're trying to build and validate an assay to the level that it needs to be, but that's how often you're seeing a positive, like, what are you comparing against? How do you control that? How do you know what the limits of your assay are? And these are really critical questions, right? And so I was approached by Slingshot, and they kind of came to me with this idea of like, we want to build synthetic controls for these kinds of situations. And as I started interacting with our CEO and our other founders, I realized that this kind of technology is incredibly powerful. Like, controls on their surface sound really boring, and then when you realize the power of them, suddenly get really, really interesting, which is why I've been so passionate about them now for the past 2 years. What Slingshot is fundamentally doing is taking many of the features that scientists care about from a cell. It could be the proteins, the nucleic acid content, the size, refractive indices, whatever, and fusing those features with some of the things that people really like about a bead technology. It's the scale, the reproducibility, the price point, and so on. And so bringing those two worlds of attributes together into our products gives us a huge amount of power to do really a lot of different things. And so Slingshot, we sell kind of standard catalog reagents that every cytometrist will benefit from. And you can connect with me if you'd like me to give you the full pitch and show you the data on why. But we also now customize, and that's probably most of our business. We build these really customized controls that fit individual assays very tightly. And especially in the cell therapy world, where you're talking about a drug product that has a protein in it that doesn't exist in nature, well, how do you control for that? How do you convince the FDA that you've made what you said you're going to make? How do you do manufacturing lot release? Because you're about to go put a cell with some artificial construct in it back into a person, and you're going to be charging them a significant amount of money to hopefully cure their disease. So you have to know if you did it or not. And the methods currently have been, um, I would say, kind of jerry rigged. It's a very new way of doing things, and there's a lot of ink being flung around over how to do it. And one of the really big powers about Slingshot, one of the things we do that's kind of been the most attractive to me, is our ability to be kind of protein agnostic, right? If biochemistry can get that protein into some kind of solution or lyo, whatever it is, if it can be isolated, there's a pretty solid chance that we can get it onto our particles. And we don't really care if that protein evolved or if it was engineered in your lab. It allows us to build things like a custom cell therapy control where one did not exist in the past. And so that's also been very exciting. That's a lot of the fun part. And I think actually that kind of is what brought me to Slingshot at the end of the day. That and the ability to do protein specific quantitation, but that's a whole other ballgame. And I see we have a LinkedIn question that popped up on the sidebar here. You mind if I give that a read, or
No, yeah,
you
you
want to
want
read
to
it
check,
to the audience?
if you've got it up, you can jump in and just read it. Yeah.
Yeah, so I can't see who posted it, but thank you very much for the question. It says, how can I assess my readiness to transition to a flow cytometry specialist role, given my experience primarily in sample running and running optimized assays and limited troubleshooting experience? Additionally, what steps can I take to gain the necessary troubleshooting skills while working as a predominantly bench-side researcher? So this is a great question, and I run into it actually quite a bit from folks who are receiving assays and receiving protocols from their superiors, from other teams, et cetera. The I would say starting point on this one would be to start asking why. Why are we using this matrix versus that matrix? You know, if you're in a clinical lab, why are we using heparin versus EDTA? Why are we using these buffers? Why are we using these colors? How do they fit together? What does it look like to design the panel? And if you are able to go and have a conversation with the people who are developing and optimizing these, folks love talking about their work. If you show up in their office and say, I'm really curious about how you got here, can you show me the data and walk me through all of your hard work here? Chances are pretty good they're going to sit down and talk to you about it. There's also a lot of really good stuff going on online. Quite a few of the instrument manufacturers have some excellent webinars, white papers, and things like that that you can read about some of these fundamental questions in cytometry. But really, again, getting to the why. Why is this panel the way that it is? Why is the matrix the way that it is? Why do I have to run it in this time frame? That will start to help you a lot with the troubleshooting. Another thing you can do is when your cytometer goes down, hopefully you have some kind of service technician, service engineer who's going to come out. Go be friends with that person. If you are able to sit down, you know, invite them to join you for lunch, talk to them if they are okay with it, right? They do have a job to do. They have metrics they need to meet, but service engineers understand cytometry in a pretty different, well, a little bit different flavor than like an end user like myself might. Service engineers can tell you so much about like how the machine is built or the instrument is built. Sorry, some labs are very particular about that. How it all fits together. What are the pieces? What are the interactions? What are the critical parts? And so on and so forth. So there is a ton out there, a lot of good resources, and even reading the papers I put in the deck, that too can tell you a lot about how to reach a desired end state for a flow assay.
That's great. And we can share that, right? So we can share those.
Absolutely. Yeah.
Fun,
All of it. It is all publicly available. I've had the very great pleasure of getting to know quite a few of the authors now, which, you know, you were subtly making fun of me for being a total flow nerd, and I copped to it completely. The first ICCS I went to, I was told by my superiors not to bring my paper copy of H62 for the authors to start signing. So it took a little while to get over the scientific celebrity starstruckness, you know, as I started interacting with some of the folks on these papers.
That's awesome. I love that story. But again, like when I come back to thinking about it, who do I want in my lab who's advising me on my controls or advising me, you know, around experiments I'm trying to create, like, you know, like I want someone that like literally thinks about it and brings a hard copy of the papers to the to the annual meetings. Like that's who I want. Who's you know, so I think a lot of times you say, well, I want to be an expert at this, so I have to be at the bench. And I think what I'm seeing with you is like you get to work with a lot of different people and work with them, a lot of different labs and companies with your expertise and with what you're passionate about.
So absolutely, and I'd say the big transition to kind of a sales or marketing career from the bench too is intellectual. It's understanding that science and everything that you are taught through your career, through grad school, whatever it is, however you got to the bench, it is a philosophy. And that's why PhDs start with PH. There is a philosophy, there is a system of logic that underlies all of it. And so, you know, I don't need to be the expert. That's what I have my customers for. They know the details. They have the expertise in what they are doing. What you do see if you take that step back, being, you know, moving from so many different projects and so many labs, you start to appreciate the basic logic that's always underlying people's experiments. And so rather than knowing the details, the emphasis actually moves to being knowing the right questions. And
Mm.
so it doesn't matter what group I'm talking to. There are specific questions that they are going to have to answer that I know I can ask them that are going to cue me in to where they are going and what they are trying to get out of their work. And so that is the fun part, right? You're constantly getting to interview other scientists who are just as passionate about the specifics of their work. You're picking up all their fun anecdotes and the crazy stories and the, oh my God, did you hear about this? Who would have thought that this thing would interact with that thing? And so when you go to the next lab that's working on it, you can say, oh, you should hear about this. And I'm thinking of all of my poor suffering mast cell scientists out there and their cytometry woes. And just, I never touched mast cells until I started working with a couple customers, and man, there are stories. So that's the cool stuff, right? You're learning all the anecdotes, you're learning all the tricks, all the little things that people have learned the hard way over time.
I like the analogy you brought earlier. That you said it was like the old peddler that would go from town to town.
Exactly.
It's actually a really interesting analogy to think of, and that again, you're bringing knowledge. Again, you're spreading knowledge not only from what you do as the company's expertise, but again, you're sharing things that across the kind of flow universe.
Exactly. And it's also really fun to build relationships with other vendors, right? There are so many wonderful companies out there, be they big or small. We're interacting with other startups all the time, learning about their new technology, talking to other salespeople, like pretty often. I have this little Rolodex in my head of like, I'll be talking to somebody. And like I was saying about making connections to solutions, sometimes people will put up a solution or a need in front of me. And I'm like, you should probably go talk to this company. I think they're working on something that would be of interest to you.
Yeah.
And I can go make that connection. And then all three of our organizations can start to have a fruitful collaboration. It gets to be a lot of fun. It's just really seeing the creativity and the collaboration take off. And then the best part is, as especially a salesperson, all of the troubleshooting you'd have to do as a bench scientist, I leave. I go talk to the next person. I come back 6 weeks later when the data is all ready. And so if you like that data high, every time I would have a big experiment complete, like doing the analysis, opening up my FCS files was like opening a giant present, and I was like, what am I going to find? So there's that data high that you get. And then as a salesperson, because you are invested in every project your customers are working on, you're just getting data high after data high after data high after data high all week, every week. And it's just awesome. Also, I have a very short attention span, which is why it's fine for me to get something like that every week.
Well, again, you know yourself, right? Like that's just
exactly
that's part of it,
Exactly.
Again, if you're somebody that just wants to sit at the bench, as you said, drink your coffee and write, or stay focused on one thing for 5 years, like that's great. That's who you are.
Yeah.
And you said something else, which I think that I say all the time is science and innovation happen through collaboration, right?
Yes.
And so if you're somebody that ultimately wants to move innovation forward, being in a role where you can recommend things or other people or other companies to people who are solving problems, because a lot of times people just don't know. Like especially when you're in a startup, or when you're in often like this tunnel vision where you're just down trying to solve problems.
Exactly.
Need somebody who come in kind of from that outside. You brought this up earlier, like understanding the bigger picture, who can say, I can, have you thought about XYZ? Because I happen to know a company that does XYZ.
Exactly. And that is I think a big thing that happened for me. I got very much stuck in my little foxhole and didn't realize that there was this whole other world going on until somebody, tapped me on the shoulder and was like, hey, if you just look up 2 inches, you'll see all this other stuff going on around you that actually could really help you. And so that was just really eye-opening. And I see that all the time. One of the other big things in my job, especially at a startup that has a new technology and a paradigm shifting, I don't know, whatever cliche you like. When you have a new tech like that, a lot of the job is actually education. And so you get a really good flavor for like how people's brains are oriented and like the lanes that they're thinking in. And then it also becomes a question of like, how do you connect with somebody based off of what they understand, and then build it to the understanding that you need them to have in order to fully appreciate your product. And you see a lot of different kinds of folks on that spectrum, right? We got our earliest adopters, the people who, for whatever reason, just took my first LinkedIn request and said, "Hey, that seems cool. Let's have a conversation." And now, pretty much all of them, we've been friends for like 2 years at this point because we just keep working together over and over. And then you have the folks that are just kind of like they're doing what they're doing and they're good at their jobs and they haven't necessarily stepped back and thought about that big picture or the underlying logic of what they are doing. And then you get folks that can be a little conservative, right? There are plenty of reasons for that. The clinical world is pretty famously conservative in that regard because the consequences of making a mistake are massive. So you cannot be tinkering around. As some of my former supervisors would say, like, you screw this assay up and you just killed somebody's kid. And that you can't play around with, obviously. But I'd also encourage folks who are in kind of conservative environments to remember that just because things are the way that they are used to doing them doesn't mean there aren't ways to make it better, right? And that's actually why I brought up the stat for the MLS techs specifically, or the MLS scientists specifically, because there are so few graduating relative to the need, the labs are having to get really creative with automation and process improvements and things like that. And suddenly, I've had quite a few really nice custom development programs where it's a clinical lab that they just don't have the manpower to go searching through, the central lab's database of phenotypes to get the controls they need for a given day. And I had one lab was spending like 120 hours a month or something for 2 people to go do that. We added all the numbers up, and I was like, that is a full-time equivalent. Like, you were literally taking 40 hours a week. It's split between multiple people, but you're still taking a full-time equivalent just to go searching for your controls. Like, why don't you just pull that out of your freezer? And so that's some of the areas that we've been able to help out a lot as well. So, end of day, time is money.
Time is money, right? And there's patients waiting. So
absolutely,
time is important. Charlie, this was an awesome discussion. Thank you for carving off your an hour of your day here for us today.
Absolutely, it's my pleasure. If you couldn't tell, really enjoy talking about this stuff, and I'm very grateful to have been able to come in and share a little bit about my observations and transition to a very unexpected career.
Well, it sounds like you're where you need to be and where you're passionate about being, and you also shed some light on again just the broad spectrum of different ways companies are using flow, and hopefully that's giving some people some ideas of where they can put a little bit of attention, depending on who they are and what's important to them as well. So I think we covered a lot today.
Absolutely.
And at the end of the day, like things continue to evolve too. So we're going to continue to evolve, and we're going to continue to have these weekly conversations with experts like yourself and people in different fields to continue to shed light on different careers that might be right for you. And so I hope maybe in 6 months or a year, maybe we have you back and we can see how the field has changed. And I'm sure it has, because things change fast in this field these days.
Constantly. Yeah, and I'm happy to even put together something a little bit more technical, delve into a particular issue if users are really seeing a lot of it, or you're getting a lot of even your questions, right? You're talking about companies coming to you and looking for specific things. I'm also happy to dig into what those things are, what they mean, and why companies might care about them.
Oh, I love that. Well, that could be some interim things we do before that next year comes.
Absolutely,
because I think there is again, I know BioTrack has a lot of people interested in their flow workshops. We see the demand growing, so maybe that is another conversation we can have to dive in a little bit more of the science and give people some better roadmaps there.
Absolutely.
Wonderful. Well, we had a few dozen people watching us today, so we can ask them what they think, and we'll poll the BioBuzz community to see what else they like. Again, as you know, BioBuzz has been around for 15 years. We've always been workforce focused, and with the launch of the Talent Lab, will now be a place where you can come to and be hired directly by employers for not only full-time jobs, but contract positions, part-time. If you're a student looking to do some part-time work, we're hopefully a destination for you as well. So again, I'm excited about the direction we're going, and what I'm passionate about is again working with people to make sure that they are living their purpose in the right career. So these events like this and talking with people like you, Charlie, us continue to fulfill our passion and purpose. So again, thank you very much for being part of that.
My pleasure. Thank you.
All right, everybody, if you have any questions, feel free to continue to ask them, and Charlie can respond on LinkedIn. And if not, that's all from us today. Have an enjoyable rest of your day, and we will talk to you again soon. Thank you.